Hi, my name is Ted Ognibene. I work at the Center for Accelerator Mass Spectrometry here at Lawrence Livermore National Lab. And I'm going to be discussing a new technology that we have here at at Livermore on the biomedical applications of accelerator mass spectrometry and doing measurements at incredibly small concentrations. AMS is an isotope ratio mass spectrometer, and it has extreme sensitivity for measuring carbon-14. And this extreme sensitivity helps us to enable studies in humans using small, realistic doses. And we use this to address important issues in both fundamental and translational research, whether that's in drug development, looking at PK or PD studies, toxicology, biomarker development, really anything. In fact, we say that in BioAMS, many compounds that we study have carbon in them. And if they have carbon, then they can be made with a carbon-14 tag. And if they can be made with a carbon-14 tag, then we can study them using accelerator mass spectrometry. And AMS helps us to establish the validity of animal models because we can do these studies directly in people. And as many of you know, many compounds fail in the translation between data in the animal models and studies in humans. And so AMS allows us to directly study the metabolism of these compounds in humans. I'll talk a little bit about the microdosing concept in humans for drug development, but essentially it gives us a rapid assessment of new therapeutics, being able to go directly into humans. Also importantly is AMS allows us to study the response from a low-level, what I call realistic exposure or environmentally relevant exposure. Oftentimes, without AMS, you have to give a human or an animal a very, very large dose, maybe the equivalent of, say, 65 million hamburgers, and then try and extrapolate down to the 1 hamburger dose. And what we find is that many times the low-level response does not scale from the high-level exposure. And so we want to be able to study the health effects at these environmentally relevant exposures. Also importantly is AMS reduces the use of radioisotopes in your research. Because of the small amount of carbon-14 that's needed and the small sample size, you minimize the risk to your human subjects and also reduces the radioactive waste disposal fees because many times the samples, after you're done with them, they can be disposed of as non-radioactive waste. I'm going to talk a little bit about the microdosing concept and also the microtracing concept. This is sort of an example of what we can do with BioAMS. Microdosing is essentially the administering of a compound into a human at 1/100th of the therapeutic dose. So it's small, and so it's not going to cause any health effects, but it is large enough that we can study the kinetics of that compound as it's going through the person. And so we're looking at administering compounds in the microgram to nanogram level. Microtracing is slightly different in that here you give a therapeutic dose to a human volunteer, but then you add a small radio-labeled component with that dose. And here you're talking about microcurie or nanocurie level doses. And what that allows you to do is you can look at the buildup of potentially toxic metabolites, and this helps address the FDA missed concerns that have been coming up regarding pharmaceutical development. The goals of microdosing is to really predict drug behavior based on limited preclinical studies and to move drugs safely into people faster. We're using this as a new diagnostic for cancer therapeutics. I think there'll be some other information regarding that. That's in collaboration with UC Davis Medical Center. And anything that with small levels of compound, such as understanding drug-drug interactions, validating preclinical models. And the example over here on the right is a study that we did a number of years ago where we dosed human volunteers with a very, very small dose of aflatoxin. This dose is essentially, it's 0.4 nanograms per kilogram. And here this individual serves as their own control in that we can look at the pharmacokinetics and the bioavailability of the aflatoxin alone, and then when it is co-administered with two other compounds, one chlorophyll and chlorophylline, to see how those compounds interact with the aflatoxin and reduce the bioavailability. This study could not have been done any other way because this compound is so toxic, and we're giving essentially the equivalent aflatoxin that the individual would be exposed to from eating one peanut butter sandwich. Our work with BioAMS is supported through our National Institutes of Funded National Resource for Accelerator Mass Spectrometry. This was established in 1999. We work with companies and universities in establishing new capabilities. We are an interdisciplinary team consisting of biologists, chemists, and physicists. And the primary role is to bring the sensitivity of AMS to the biomedical research and the business community. And we have a number of collaborations with both universities and businesses ongoing now. And our website here is listed at the bottom of the slide. I talk a lot about the sensitivity of AMS, and here I want to prove it to you here. Only AMS offers proven part per quadrillion quantitation. Now, part per quadrillion is 1 part in 10 to the 15. And what I've listed here is a bunch of other commonly used analytical techniques in the biosciences for quantitation of compounds. And these are the typical sensitivity ranges of this instrumentation. And you can see that only AMS gets you down to that 1 part in 10 to the 15 level. No other instrumentation comes even close. However, we're extending that sensitivity even further with our liquid sample AMS capability, and now we're getting down to the zeptomole, which is the 10 to the minus 21 mole range. And this interface solves a very critical need that we had with sample preparation, in that prior to this instrumentation, the sample preparation took relatively dexterous handling and a couple of days to process. And we've been able to realize a significant reduction in the minimum sample size in the carbon-14 content. So we're able to reduce the sample sizes down to about 10 nanograms of carbon, which is a 50,000-fold reduction. Carbon-14 sensitivity is down into the 50 zeptomole range, and that's a factor of 20 reduction in the amount of carbon-14 that we can quantify. Importantly, the analysis is done in minutes as opposed to days. You can put a sample down onto the interface and measure your result. Basically, you get a number back in about a minute. And this enables directly coupled HPLC AMS analysis. Prior to this instrumentation, you would collect fractions and then treat each fraction as an individual sample, again, taking several days in order to get your data. Here we're able to directly couple the output of that HPLC to the quantitation afforded by the accelerator mass spectrometry. And this leads to a significant reduction in the per sample costs, as well as the complexity of your measurement. And we have a number of opportunities that exist for collaborations and other fee-for-service activities. My name is Ted Ognibene. I'm at the Center for Accelerator Mass Spectrometry. If you have additional questions, you can contact Kevin Christopher at the lab's IPO office, and his email address is listed there. Thank you.